So good evening, everyone. Uh, in our last class, we have talked about the instrumentation behind the single beam spectrophotometer. Okay, and we have seen that in a single beam spectrophotometer, we have a light source, then we have a collimator and the prism grating. Okay, so this collimator it concentrates the light which are being incident from the light source onto the prism, and then this prism or the grating it breaks down the spectrum into different color bands, and then the wavelength selector what it does is that it selects only one specific wavelength of the light, and that is it makes the uh, incident light onto the sample a monochromatic light. Okay, and then that monochromatic light it passes through the sample solutions and it falls onto the detector, which is then being sensed by the detector and then it is processed. So the amount of absorbed incident radiation which is being generated. So over here, say if it is I0, and then some of the these things are absorbed, say IA, and some of them are transmitted IT. So so depending on the amount of the transmitted light, the IA may be calculated and the absorption by the solution is calculated, and that information is used for analytical purposes. Okay. In a conventional split beam spectrophotometer, we have discussed that there are two types of lamp, D2 lamp and tungsten lamp. Okay, the D2 lamp it is used for the generation of radiations in the UV range, whereas the tungsten lamp it is used for the generation of the radiations in the visible range. So combining these two, we get the UV visible spectroscope. Okay. So when the scanning is being done in the UV region, this D2 lamp is on, and then what happens? Uh, the light from this is being incident onto the mirror, and then mirror reflects the UV radiations onto the monochromator, which is having an entrance slit and an exit slit. Okay, so through the entrance slit, the UV radiations they fall onto a monochromator, which is basically the gratings. Onto the gratings, it then what you call it then decomposes the UV radiations, and then from there, from the exit slit, only the radiation of a specific wavelength they are being emitted out, and then we have a beam splitter which splits the beam into 50% where the 50% of the UV radiations are being transmitted and 50% is reflected. Okay, then one of the among the two 50%, one goes through the sample, which sorry, one goes through the reference. Okay, and one goes to the sample, and both these transmitted lights from the reference and the samples they are being compared, they are being detected by the photodetectors to independent photodetectors, and then the data is processed, these transmitted lights are compared, and accordingly a um, what you call spectrum is being generated. So once the scanning in the UV is done, then the visible light it glows on, and the D2 lamp is switches off, and the process is being repeated. Okay, and by this way we can get a total UV visible spectrum. So this we have discussed in our last class. So up till now, do you have any problem? No.
No,
Good.
sir.
Okay. Yeah.
No,
Another
sir.
important thing which I forgot to mention over here, in the single beam spectrophotometer, if this collimator, prism of grating, and the wavelength selector, if I combine this and say if I have this lens, this collimator lens, and if I color it, so it becomes an optical filter. Okay, it becomes an optical filter, and what happens over here is that this optical filter it converts the this your polychromatic light into a monochromatic light. Okay, up to this region they they are being stopped, and they are only a single lens is used, color optical lens is used, which is basically an optical filter. Okay, so this lens is now being used for converting the polychromatic light to the monochromatic light. Okay, now there is one problem over here. What is the problem? For specific wavelengths, you have to get specific filters. So the number of filters uh, which can be what you call included within the colorimeter, that is limited. So usually 7 to 10 number of uh, optical filters they are included within the colorimeter. Okay. Now this is the schematic layout of an image processing based spectrophotometer. Okay, and this is a very uh, simple type of spectrophotometer which can be designed in our lab. Okay, and slowly the companies they are moving towards this kind of spectrophotometer uh, due to various reason. One, if you look over here in this split bin spectrophotometer, you have a this grating which have to be moved. Okay, so there is a rotational element. Okay, you have to rotate this gratings so that specific wavelength of the radiation it falls onto the exit slit, and only that radiation gets out of the this monochromator. Okay, so there is a movement of these things. So your what you call so there is a lot of movements over here. Okay, now due to this reason what happens is that if something gets out of order, then getting this monochromator fixed, you need specialized manpower who can rectify this thing. So the downtime, if the machine undergoes a breakdown, so the downtime will be quite high. So if you look into this image processing based spectrophotometer, um, it has got a all the things they are fixed. The first one is the mains. From the mains, it takes the power. So you light a radiation source. Okay, then it falls through an entrance slit onto the sample. Okay, and through here, through the exit slit, the radiations come out. Now please look over here. Once the radiation is being generated and the radiation which is getting out of the sample, they are polychromatic radiation. Okay, in the previous cases, two cases, both in the single beam and the split beam spectrophotometer, the radiation which is incident onto the sample, it is monochromatic because we are using a monochromator. Okay, so here we are incidentating a monochromatic light onto the sample. Okay, whereas in the image processing based spectrophotometer, we are incidentating a polychromatic light onto the sample, and we are getting a polychromatic output light. Okay, now this slide it goes, and if you look in this green area is the grating. Sorry, today it's a bit cold here, so my fingers are getting frozen. Uh, that's why my writing is a bit wonky, so please bear with me. So the grating, this green part, it is the grating. Now, what does the grating does? If you look in our previous case, what is the grating doing? It is breaking down the polychromatic radiations into a band of radiations. Okay, so it is decomposing the light. Okay, and the same thing happens over here also. This grating, it will decompose the light, and then that decomposed light will be incident onto the camera, okay, which is basically an array of diode or CCD sensors, or even maybe you can have CMOS sensors. Okay, then that image would be captured by the laptop or the computer or the processing unit, and you can do the processing over there. Okay, so this device, it was developed by one of our student in our lab only. Okay, since I am in the medical electronics and instrumentation lab, so the student has devised this thing, and through that he has captured some spectrums. So if you look into the spectrum, what is this? Can you see? It is just breaking down the different components of light into different bands. Are you getting my point? Now, the first one, it is the reference this thing. So what we have designed over here, if you see, we have designed a single beam image processing based spectrophotometer. Okay, so in the single beam, first we have to take the reference. Okay, so he has taken the reference, and then foot color. Here we have used orange red foot color that contained sunset yellow, FCF, and carmosine colors. Okay, so orange red food color which are available commercially in the market, it was taken and it was dissolved in various concentrations. The concentrations have been given over here. Okay, so if you see, as we go from left to right, the concentration increases. Now if you see in the first case, there are certain blue components which are there. Then in the second one, in the C component, as we increase the food color dye, the blue component was present, but its intensity was reduced. And in the D and the E, there were no blue color components. Okay. So on the contrary, if you also if you see the green color information, it was very bright initially, then it reduced, then again it reduced, then again it reduced. Okay, so you can see how it is behaving. Now if we go back to this color wheel, if you see, we are working in this region. So while discussing this color wheel, what we discussed is that this orange color, it is complementary to the blue color. Okay, so if I have to block the blue color, then I have to use an orange color filter, right? Now this is what you are seeing over here. Now when you are putting these solutions, food color solutions, into the sample slot over here, so we are having the radiation source which is coming, and then through this sample it is falling onto the image sensor through the grating. So this sample, this 6 point, this sample, it is acting as a filter, right? It is acting as a filter, and then you see as the concentration is increasing, your blue color components, they are decreasing all throughout. Okay, it is decreasing all throughout as the concentration of the orange red food color is increased. Okay, now from here, how do you find out the spectrum? So what you do is that this is the image what you get from the camera. Am I right? This is the image what you get from the camera. Now, for your typical source, the spectrum, if you do not induce this filter, include this filter, this light will be incident onto the grating, and then the image will be formed. So you will be for your this thing for your source, you'll be getting an image like this. Now, once you have got the image like this, you can compare spectrum of the what you call of the source with the what you call standard pattern which you get. Okay, so once you get this image, you draw a you take what you call not draw you basically take the pixels which are in a particular line. Okay, in our case what we did, we took say 4 to 5 layers of the pixels over here, and we did the we got the readings. So what I am talking about, I'm talking about say so like say you'll be getting like this thing, the pixels will be there. Here I have shown phone number of the pixels in a column. Okay, now what you do is that this one column, it represents a particular wavelength. The second column, it represents another wavelength. Okay, so you average this down, average these 4 cells, and you get a single row like this. Now, what is there in the x-axis? In the x-axis, you have pixels. Okay, and what these pixels have? These pixels have intensity values. Right? So in x-axis you have pixels, in y-axis you have intensity values. Right? Now from here we can plot a graph of intensity versus pixels. Okay, pixels mean pixel positions. Okay, so the first position will be 0, 1, 2. In this way it will go on, right? So here also pixel position. So you will be getting the spectrum of the light. Now you know what source you have used. So these positions you will be knowing. Now since you will be knowing these positions, what you can do, you can map that, you can find out that this position is at say pixel number 10. This position is at say pixel number 20. Okay, so accordingly you try to map the you convert these pixel positions into the wavelengths. From a known source, what you will be getting? You will be getting wavelength versus intensity. Okay, so for a known source, you'll be getting like this. And you know this lambda 1. What is the lambda 1? This is lambda 2. This is lambda 3. So you can calculate this 3. And from here you have the pixel positions versus the lambda positions, and you can get the information. Okay. So this is how you can calibrate the sensor over here. Okay. Now once you have done that,
Sir,
yes, please.
sir, how that length was determined?
Beg your
What
pardon,
will
I am.
happen if you
What will happen
if you will increase or decrease that length? That length was
which length?
140 in an
Yeah.
sir last page.
Okay, so this you have to standardize. Okay, this you have to standardize on your
Yes, sir.
own, because you have to make the this thing, your the decomposed spectrum fall onto the image sensor. Okay, because you have to do the imaging, right? Now when you are doing the imaging, you have to make sure that your spectrum is incident onto the image sensor in a proper manner. For that thing, I will come to this slide. Okay, if you look into this grating, okay, so what is happening is that when your sample is being incident, so what happens over here is that if you place your detector, in this case the image sensor, closer to the grating, if you see the distance or the image which will be found will be in a smaller region. You're getting my point? So what will happen? You
Yes,
have the
sir.
same value and everything is in a very congested manner. All the wavelengths are in a congested manner. Now, if you take this detector slightly towards the right, what will happen? The same spectrum will be incident on a larger area. So, the resolution of your uh, what you call wavelength detection, it will increase. Okay, similarly, so what is grating we have put over here and we have found we are getting good result. And when you talk about this thing, this sample holder, it is acting as a filter. We have already shown that it is acting as a filter. Now if you remember from our single split this thing, what this detector does, what this filter does, it tries to what you call make all the incidents, all the lights incident onto the grating in a concentrated way. Here also you have to find a fine sweet focus point. Okay, because we are not using any filter over here. I told it is acting as a filter, but it is not an actual filter. So we have used an exit slit over here at 7, right? So a narrow beam of light is coming and it is falling, but falling onto the grating. If you move this grating far behind, then what will happen? There are chances that this what you call the spreading of light may happen, and if the spreading of light it happens, then it will degrade your image quality. Okay, that is one of the thing. Then you can play with the positioning of the grating depending upon your image sensor. Okay, am I clear?
Okay. Yes, sir.
Okay. Now once you have done this, you see at the lower wavelengths we are having the blue components are there, And as we have increased this blue component, see, it has decreased. Here it has been completely suppressed. Here it has been completely suppressed. Okay. Another important thing, if you see the intensity of this one, see 150, it has increased over here. It is increased further over here. It has further increased over here. Okay. So what is the last peak? If you see the reddish color, it is showing an increased intensity. The initial one, there was a lower intensity, and as you go on, you get a larger intensity. Okay. So you, if you not have a spectrophotometer, visible, if you are working on the visible spectrophotometer, you can design your own spectrophotometer in your lab. Okay, it will not take you much amount of money. If you have a basic knowledge of the electronics, you can design your own spectrophotometer. And as a teacher of the medical electronics and instrumentation, uh, I would guide you in the following lectures. Whatever the techniques we study, I will try to give you some idea. If you do not have any costly equipment, how you can make a roundabout and you can try to get your things done in a what do you call, if in Hindi it's called jugaad. How you can make a jugaad and get your work done. Okay. So this is how you can develop your own spectrophotometer in your own lab, and you will find a lot of videos, YouTube videos in the YouTube also. Okay. Now coming to the UV visible spectrophotometric detectors, we have phototube. Second one is the photomultiplier tube. Third is the diode array detector, and fourth is the charged coupled devices. Okay, these are also regarded as CCDs. Okay. These two, these are used for imaging based spectrophotometers. Phototube and this one, these are used for conventional spectrophotometers. So phototubes comprises of a light sensitive cathode and an anode inside an evacuated quartz envelope. Why quartz envelope? We have discussed that the quartz envelope, it can allow both the UV light and the visible light to pass through it. And since we are working with the UV visible spectrophotometers, hence the phototubes which we will be designing, they should allow the photons or the radiations from both the UV region and the visible region to fall onto the photocathode. Okay. Photomultiplier to the most popular detector used in UV visible spectroscopy. It comprises of a photosensitive cathode, anode, and several diodes. Basically, this photomultiplier tube, it is an upgrade of the photo tube. Okay, so herein the sensitivity is quite high. So here we have shown the working principle of the different uh, detectors. The first one is the phototube. The radiation or the photon, it falls onto the cathode. Okay, from there the electrons are being generated, and though that electron, it is being accelerated towards the anode, and from there due to this, the movement of this electron, there is a generation of the current, and that current is being detected, and that is being sensed. However, the sensitivity of the phototube, it is lower. Hence, what happened is that this photomultiplier tube, it is being used. So what happens over here? The radiations, it falls onto a scintillator. Now this scintillator, it generates a couple of electrons. So one photon will generate at least 4 to 5 of, photons. Okay, that those photons, they will be exciting the photocathode. Now that cathode will further generate some more electrons. It will generate some electrons, and those electrons, it will move from the photocathode to the diodes. And each of the diodes, if you see, they are connected with the cathode of the power supply. Okay, now at each diode there is a multiplication of these electrons, and as mm, these electrons reaches the anode, this anode plate, the number of the electrons have been multiplied quite significantly. Suppose if you see here, it was only one electron which has come. So from here, one electron came over here. From here, when one electron resulted in the generation of the two electrons. From here, two electrons resulted in the generation of, say, three electrons. From three to four, and so on. It went on. Okay, so there was a multiplication of the electrons which are being generated, and at last, when the electrons reach the anode, a larger amount of electrons, they entered into the electronic circuit, so a larger amount of current has been produced. Okay, so due to this reason, this photomultiplier tube, they are more sensitive as compared to the photo tube. Okay, then we have diode array detector. So this is a schematic representation. So what happens? You have a array of diodes, p doped silicons are there. This darker shades, this thing. Then you have n doped silicon, and in between these two, you are having depletion region. Okay, and all. I hope all of you know how the photodiodes are being arranged. Okay, so I'm not going into the details. So basically, the photodiodes, they are being put in the reverse bias conditions for their proper functioning. Okay, so what happens from the gratings? If you see from the gratings, the radiations are being what do you call, the radiations have decomposed, and once they have decomposed, and as you bring this detector, take this detector away from the grating, what will happen? You will be having good resolution. Okay, however, if you take it too far, then what will happen? There will be a diffusion of the light, and there will be beam broadening and all this thing, so which we do not want. So there is a sweet spot where you can keep this detector so that the radiations are captured in a proper manner. Okay, so this arrays of the detector, it helps you to get the information uh, in a spatial domain. So if you see from here to here, we are in the spatial domain, but when the photons or different wavelengths are striking, we are basically converting this distance information into the wavelength information. Okay, and for a particular source, the wavelength component should be known, and you can break it down into you can convert this distance formula to the wavelength. Yeah. Okay, you can correlate it. Okay, so from there you can get the wavelength versus intensity plot, right? Similarly, we have the charge coupled detectors. So in the charge coupled detectors, you will be having a series of this charge coupled detectors. Okay, so they will be helping you out in getting the um, in capturing the photons in an efficient manner. Okay, so as compared to the this diode array detectors, the CCD sensors, they have higher quantum efficiency. They have higher quantum efficiency. Okay, and which may reach up to 90%. Okay. On the contrary, when we talk about our eyes, they have a quantum efficiency of nearly 1%. Okay. As compared to the diode arrays, these charge coupled detectors, they have better quantum efficiencies. Okay. However, these charge coupled detectors, they are slower in response, but they have a very high signal to noise ratio. Okay. Nowadays, another type of imaging sensor, which is regarded as CMOS sensor, they are being also being used. Okay. However, the CMOS sensors have a lower signal to noise ratio as compared to the CCDs. They have lower quantum efficiency. Okay, as compared to the CCDs, but they are much faster than the CCDs. Okay, in CMOS, you will be getting the CMOS sensors over there in the array form like this way, and each of the sensors, they will be having their own amplifier. However, in the CCDs, it is not the case. In CCDs, say they will be having a set of mm, amplifiers which will be used for a specific row of the CCD sensors. Okay. However, the CMOS sensors, they have an amplifier for each of these CMOS sensing element, and this is why the CMOS sensors, they are very fast as compared to the CCD detector. However, um, on a the CCDs have various advantages, like they have very high quantum efficiency and they have high signal to noise ratios. But as the days are progressing, the CMOS sensors, they are catching up with the SNR and the quantum efficiency of the CCDs, and in near future you will find the CCDs will be what you call they will be completely eliminated by the CMOS sensors. Okay. Now coming to the sample holder. In the sample holder, you have UV visible spectrum. If you want to do the UV visible spectrum analysis, you have to use either quartz or fused silica as the sample holder. Then, if you are only interested in the visible spectrum analysis, you have silicate glass and acrylate cuvettes. Okay. Then you have cuvette size. Most of the cuvettes are designed to fit into a 1 centimeter square holder into the instrument, but as per your requirement, you may have a large number of cuvette sizes. Okay, so if you look over here, we are having 1 mm by 1 mm to 10 mm by 10 mm. This 10 mm by 10 mm, it is it is basically a square like thing. Okay, so 10 mm by 10 mm. Okay, and in the all other things also you can have this stuff. Now, why you need a longer or a broader this thing cells? The reason is that you want a broader cell due to the reason that if you have a dilute solution, then the concentration will be quite distributed. You will be having very low concentration. Now, this using a very large cuvette, it will allow you to have a measurable amount of absorption. Okay. However, when you go for this higher absorption, the signal to noise ratio may reduce. Okay, signal to noise ratio may reduce. That means your noise component will be higher. However, that can be taken care of if you measure the spectrum for a longer period of time and then averaging it. So usually what happens, suppose if you want to do the find out the signal to noise ratio of an average which has been averaged for m times, then it becomes under root of m of the SNR. Okay, so the more you do the averaging, the lesser, the higher will be your signal to noise ratio, because it is being getting multiplied. This under root m, it is getting multiplied. Okay, so the so the long here now what you have, you do not have to do the multiple number of things, but if you expose it for a longer time and you then record it at different instances and then average it, then what it will do, it will help you in getting the good signal to noise ratio. And this is where, if you have to do this kind of analysis where you have very dilute concentration of the analytes, the CCDs come into place. Okay, because CCDs, uh, since they are having very high quantum efficiency, even a small change in the intensity can be easily picked up by the CCD, and it in its inherent mechanism, how it detects the photons, they themselves have the inherent mechanism of the averaging for a longer period of time. Okay, now here some of the measurement wavelength range for the different types of cuvettes, it has been given. So please look into this. So you will find in the cell culture many people they use plastic like cells. They are either polystyrene cells or the polystyrene cells or the PMMA cells, polymethyl methacrylate cells, and they are only used for visible spectroscopy. These two types of cells, they cannot be used in UV spectroscopy. I have seen students that they come with these cells and they try to do UV visible UV spectroscopy. But please be reminded, if you are having any plastic cells, plastic touch, feel like cells, okay, that might be the polystyrene or the PMMA cells, they are not to be used with the UV region. If you want to do the UV analysis, you cannot use this these two cells over there. Okay, if you want to do both UV and this cells, then you have to go for both in the UV and the visible region. You have to go for the glass cell, quartz cells. Okay, glass cell, it is regarded as G cell, quartz cell, it is regarded as the S cell, then IR cells, it is having an extended region up to the IR spectrum. Okay, so these are the different types of cells which are there. Okay.
Sir,
So,
is
sir,
it essential to have?
go ahead, Ganshi.
Sorry, I couldn't get you. It is essential to have.
Sir, I was asking, like, is it essential to have the one surface of this cavite opaque or something? Because in the genetic engineering lab, in the video, I saw that thing.
Okay, you are telling that why two surfaces? Because if you consider this thing, if you look into the top side, so suppose your source is here, so this side and this side are made transparent, and over here you are having the two sides which are grounded, right? And they are opaque. Am I correct?
Yes, sir.
Yeah. So these are done on purpose. The reason being, if you have seen that this side and this side, they are transparent. Okay. So when you are putting into the this UV visible spectrophotometer, you have to make sure that this is in this direction, and these two surfaces, these two surfaces which I am marking as red now, these two surfaces you are not supposed to hold the cuvette through this surface. Okay, what happens is that this is a very important question what you have asked. I have seen many students, what they do, they hold the cuvette through these two sides. So when you do that, if you have studied the anatomy of our body, in our fingers, we are having small pores through which sebum comes out, which are basically fats. Okay. Now these fats will be transferred onto this region. Now these fats will be transferred and you will be having fingerprints over this region, and you don't want that. Why you don't want that? Then what it will do? You are analyzing the sample which is present within it, within this cuvette, right? So if you make a light incident on that, these fats they will also absorb the some of the radiation, and the signal what you will be getting out of these cells which are falling onto the detectors, that will be an erroneous result. So in UV visible spectrophotometric cuvettes, you will find the two sides are being grounded, and you are supposed to hold the cuvette through these two sides, to these blue sides, okay, not through these red sides. If you do this, then you are damaging your cuvette over time, and it is very difficult to get rid of this fat. Then you have to go for this aqua regia and all this thing, chromic acid solution. You have to make chromic acid solution. You have to put those over there, and it takes a lot of time. Okay, so ultimately you have to do, but if you can um, avoid this, that will be better, because then you have to clean the cuvette after a long time. If you just rinse it and then dry it, that should be okay for you. And if you touch these sides by mistake, then after every time you have to clean the cuvette using this tedious process. That is why two of the sides are making are what you call, they are grounded, uh, to make the people understand that these two regions, they should be put into the optical line of sight, LOS, we call it LOS, optical line of sight. Okay, and while holding, we have to hold the grounded through the grounded parts. Okay, or the grounded sides, not parts, sides. Okay, anything else?
Yes.
So normal glass is opaque to UV light.
Yeah,
Okay. Okay, sir.
I have told you previously also.
Yes, sir.
If you look into the silicate glass, I think monochromat, somewhere I told you. Yes,
Okay, yes, sir.
silica
Yes,
glass,
sir.
silicate glass.
Yes, you told us.
350 nm to 3000 nm.
3000
So
m.
these are opaque to UV.
Okay, sir. Okay, sir.